Good afternoon, everybody. We're going to get started. My name is Ken Bloom. I'm Chief Medical Officer of Clarient, part of GE Healthcare. I'm going to talk about some exciting things that we've got going on within our organization. A couple of housekeeping things. Some of the things that I'm going to talk about, we've got articles in the back of the room related to the references on those things. And I'm happy to stay behind if people have questions, if we go over and anybody wants to ask questions, more than happy to do so. I also have to apologize for Dr. Allred, who is sick with the flu. So I'm going to take his part, sends his apologies. So Clarient is a company that's been in existence since 2004, and its mission statement was to improve the lives of those affected with cancer by bringing clarity to a complex disease. The premise when we started the organization back in 2004 was really to bring tools to community pathologists. The idea was that at meetings like these, we were going to hear about all sorts of new molecular diagnostic tests that might be outside of the reach of the local pathologist, and that Clarient would be the company that would provide those services so that everybody would have equal access to health care throughout the United States. And we did that really by providing an infrastructure in which we could do some of the testing and return those results back to the community pathologist. And we've always held the vision of the community pathologist as being the key integrator of the information services that we provide. Now, I've been doing breast pathology now for going on almost 30 years now. It's hard to say that. But, you know, our good old histologic variables that we look at still have a major place in the day-to-day practice of pathology and certainly in the care of breast cancer patients. And sometimes we lose sight of that as we get enamored by the new technologies, and I'm going to describe some of those new technologies today. But you have to remember that the basics upon which we're grounded are the key pathologic variables that are routinely done by the pathologists in your community, things like tumor size, the subtypes of cancer, tumor grade, lymph node status, vascular invasion, et cetera. And you know, we all know in our practice that there are certain subtypes of breast cancer that have exceedingly good prognoses, with greater than 90% survival at 10 years. And it's very difficult to come to a conclusion that we should be aggressively treating those subsets of patients. And we see that reflected now in guidelines, even in things like ER and HER2 guidelines, saying that you have to at least consider histology in the interpretation of the sorts of more detailed molecular analyses that you're undertaking. And again, if we just look at the key variables like tumor size, grade, and lymph node status, and plot that in a Nottingham Prognostic Index, you can see we can predict fairly well, almost as well as most molecular markers today, in how patients are going to perform. We've extended that out to the key biological markers for therapy, things like ER and HER2, absolutely critical variables, but the key thing about these markers are that a bad result on those tests is as bad as the drug not working. So you have to have an appropriate test, which means good quality tests with good quality observations by the pathologist, so that you get the right result to make the right decisions in treating your patients. And in large part, that's what Clarient was about. We recognized the difficulty in performing some of these tests accurately, especially in small laboratories that might not have sufficient volume to adequately validate those markers. And so we served as that repository, as well as bringing and providing guidance to the pathologists in interpreting those results. We've expanded out our breast team at Clarient. So Clarient started in 2004. It's a relatively small organization. But over the last 8-9 years, we've acquired several experts in breast pathology. I was one of the founders of Clarient. More recently, we've brought on Neil Goldstein, Noel Widener, and Craig Allred. Many of these names should be familiar to you. They've all published literally hundreds of articles in breast prognostic and predictive markers, as well as breast pathology by itself. So we've all learned through these meetings and through the literature that breast cancer is no longer considered a single disease, and no matter, we knew this back actually 30 years ago when we graded tumors and we looked at them and we said, you're a tubular cancer, you're low grade, you're going to do very well, you're a very high grade 3 tumor, vascular invasion, et cetera, you're going to do poorly. Now we can do the same thing with molecular subclassing. We can do it by looking at the genome, complexity of the genome, as well as a variety of other techniques, next-gen sequencing, et cetera. And of course, we're all familiar that you can subclassify by gene expression. Chuck Peru's work, and repeated by many others, showing that gene expression can classify various subgroups of breast cancer with different outcomes. Again, very similar to what we could do with morphology. More recent publication a year ago, followed up with genomics earlier this year, the Metabrick study broke that classification down into 10 different subgroups, each having different prognoses. So really, it's just whatever methodology you're using, you can start subclassing breast cancers into those that have a more favorable course and those that have a worse course, with full varieties in between. And I'm going to talk a little bit more about that later on. But precision pathology is the future. Just looking at something under a microscope, we knew how far we could get, but we were still not very good at predicting on an individual patient basis who was going to recur and who wasn't. We were great in populations, not so good with individual patients. And this quote, more than 100 years ago from Sir William Osler, sort of summarized it all up. If it weren't for the great variability among individuals, medicine might as well be a science and not an art. And for the pathologists in the room, we all know that when we look at slides under the microscope every day, we want that to be more and more precise, but there's an art to it. There's an experience level that just plays in. And we know that not every slide looked at by every pathologist gets interpreted exactly the same way. So we would like to make things more precise. At that same time, the job of the pathologist is changing. So I like to say when I started in breast pathology, life was easy. There was no such thing as a breast pathologist. It was sort of the throwaway surgical pathology rotation. That's all you had to do was size, grade, and lymph node status. That was pretty much it. We would cut specimens in. They'd sit in formalin tanks for a week or 10 days before we looked at them under the microscope. And the sort of decision points that we had to make were relatively straightforward. But nowadays, there's lots of decision points that get made off of what we do. And it's really a fairly substantial subspecialty nowadays. It's very difficult to be a generalist pathologist and keep up to date with all of the nuances of breast pathology. The sorts of things that we have to answer has changed, though, too. So it was always, what does this diagnosis mean for the patient? Should they have a mastectomy? Can they get away with a lumpectomy? Do they need radiation therapy, et cetera? But now, it's really, how do I intervene to change the natural history of that disease? Can I figure out what's driving that tumor? Can I target that? Can I figure out what the resistance mechanisms are going to be so that you can actually develop a more holistic treatment plan around the patient? In addition to that, families are now very interested. So now we can delve into the genome and understand the molecular underpinnings of some of these diseases. The question arises, is my family at risk? Can you identify something that might be important to them? And to answer these sorts of questions, new tool sets are required. So in the 80s, it was great for me to answer all of my questions with just my microscope, but now things are more complex. So some of the things that I have to quantify, I now need tools like image analysis to help make that more precise. I need to be able to combine methodologies like IHC and FISH to get a better understanding of what's going on in individual cells. And of course, we all know that next genome sequencing is going to add a whole host of new markers to our decision-making process. Now, this is an article back from 2007. A group of experts in breast care got together and tried to prioritize the problems in translational research. And in large part, these are problems because we're not quite sure how to treat individual patients. These are all questions that are directed at, what do I do with Mrs. Smith instead of a larger cohort? So at Clarient, we were initially organized predominantly around diagnostic services and bringing that technology to our local community pathology partners. But in 2010, we were acquired by GE Healthcare, and that acquisition by GE Healthcare brings the whole gamut of GE power behind it, including all of the in vivo technologies and all of the life technology technologies, as well as the information system technologies that existed within GE Healthcare. And Clarient sort of resaw itself to saying, yes, we provide these diagnostic services, but we're also now going to advance into genomic services, and we also have a significant part to play within the biopharm research area to develop these next generation tests that are going to become important to providing individual care for patients. And as we all know, there's significant challenges in these research. Breast cancer is a complex disease, right? It is not involved just a single pathway. Multiple pathways are involved. Heterogeneity is a big issue. And while we like to think of things as expression analysis and next-gen sequencing as being important tools, when you layer on the heterogeneity within individual breast cancers, and I'm going to show you some data around that, it sometimes obscures what's actually going on in individual cancers. And of course, we don't want to become a single platform company and say the only thing we can do is next-gen sequencing or the only thing we can do is expression analysis. We're a full-service laboratory, and so we really don't care whether the marker turns out to be a protein marker or a genomic marker. So our goal is really finding clinical utility. I like to not call it personalized medicine or even precision medicine. Really what we're providing is stratified medicine. We're still not taking care of Mrs. Smith. We're just getting smaller and smaller cohorts of patients that are similar to each other to try to treat them in a similar fashion. The premise of Clarient from the get-go has been about really being agnostic to platform. So it's really choosing the right platform for whatever the right question is. So we're agnostic to platform, apply the appropriate bioinformatics, and now with the GE solution, try to integrate the in vivo and in vitro data to come together with a single solution to the patient. So simple things that we did early on, trying to get reproducible scoring around HER2, for example, seems easy, but if you call that 3+, then the patient gets treated. If you call it 2+, then you're going to do additional testing to see whether patients are eligible for therapy. And if I polled the pathologists in the room, I would probably get mixed results on a slide like this, some calling it 3+, some calling it 2+. And so the idea was, could we apply more standardized tools to help solidify that decision process? And so things like image analysis to help separate out tumor from non-tumor, eliminate overlapping cells, and try to get reproducibility around where that staining is explicitly on the membrane and the intensity. And so we can use tools to codify those cells to say some cells are 3+, some cells are 2+, develop ratios to say does it reproducibly meet a criteria for therapy or not. Same thing with in situ hybridization testing, whether it starts out with fluorescence in situ hybridization or moves more now toward the chromogenic in situ hybridization. The idea is to bring expertise and to train pathologists on how to interpret these slides accurately and reproducibly. Now, more recently, we've delved into subclassifying cancers on the basis of genomic analysis. This was published last year from the Cancer Genome Atlas Network. And I think what was interesting about this, at least to me, was that there were very few genes that were very prevalent within breast cancer, other than P53 and PI3K. The majority of other genes that were identified were prevalence levels, south of 10%. So really what it said is that the sorts of molecular alterations that we're seeing in breast cancer are actually quite unique to individual breast cancers. We're not going to have large cohorts of patients showing the same abnormalities. And in addition to that, the other finding that was interesting is the most common alterations, or near the top of the most common alterations, the PI3K alteration was actually most commonly associated with the tumors that had the better prognosis. So that was an interesting finding, sort of opposite of what you would typically think, that all mutations should be bad. Some mutations might actually be better. And in a recent publication earlier this year, they went back to that Metabrick analysis that looked at the expression profiling into the 10 subgroups. And when you apply and overlay the molecular data on top of it, I think the thing that gets highlighted to me in this is the tumors that do better have less genomic abnormality or less genomic stability. But the ones that do better have more genomic stability, fewer overall mutations than those that have a poor prognosis. So you can almost look at genomic instability as a measure of whether this tumor is going to behave in a fairly good fashion or a relatively poor fashion. The other thing that complicates the question with doing molecular analysis is the intratumoral heterogeneity. So this is a renal cell carcinoma that was published in New England Journal about 2 years ago now, in which they took multiple sections of the same tumor as well as several of the lymph node mets and did complete genome sequencing on each of the lesions. And what they found was interesting was that in gray you can see where they found mutations, and about a third of the mutations, a little bit less than a third, were in all of the tumors. It didn't make a difference where you sampled the tumor, you would find that particular mutation. But there were subsets of mutations that were only found in the primary but not the metastasis. There were other tumors that were found, other mutations that were found only in the metastasis but not in the primary. And then there were what we'll call personalized or private mutations that were just in one section but not in any of the other sections. So very intriguing by itself. So what does that mean? Well, if only 31% of those somatic mutations were ubiquitous, then it meant that the profile that you get back with your results is dependent on which block you selected for the analysis. And that becomes very problematic because you don't know which gene is actionable. Beyond that, we're not sure which one of those are actually driver mutations and which one of those are passenger mutations. And so while this is very exciting from a research standpoint, it's very difficult to apply to clinical practice and say, what specifically does this mean for Mrs. Smith, for example? And for the pathologist, it really presents large problems because I just get my breast tumor, and now how I select the block will determine what findings you actually get, which will dictate the treatment. So if I select my block differently, the patient might actually get treated differently. That's a scary thought as a pathologist. So we want to have deeper understanding of what's actually going on. And we have to be able to translate this to pathologists as a whole to say, this is the standard way of taking a breast specimen. For those of you that don't know, the way that we typically take breast specimens is predicated from really 50, 60 years ago, which is you take a section of what looks like tumor to confirm that your gross observation of what looks like tumor was. You take sections of normal to confirm that what looked normal to you actually is normal and doesn't contain tumor. And then we would take interface sections so that we could assess margin status. So we weren't taking them with the idea of what's the best way to take sections to assess biomarkers. So that's something that's out there. And I think as a group, as pathology and actually radiology as well, it means we're moving toward more integrated diagnosis. We have to figure out how we're going to merge technologies to understand that what we see visibly that leads to that biopsy might help lead the pathologist to select the right area of the tumor to do deeper molecular analysis. So to get into that game, we purchased a company known as Seqrite in Houston. They were established in 1994 as a DNA analysis company, so they've been doing this deep analysis for over 20 years. Doing all sorts of molecular analysis from next-gen sequencing to Sanger sequencing, quantitative PCR, microarrays, huge bioinformatic pipeline support, as well as custom molecular biology. And one of the things that was very appealing about this company to us is they were very similar to us in that they were platform agnostic. So they were not just a sequencing company or a molecular company that had one toy and they were playing with that toy. In point of fact, they were very deep in doing Sanger sequencing, quantitative PCR. They had Affy arrays, as well as the whole gamut of next-gen sequencers. So MiSeqs, HiSeq, Solids, 454s, IN torrents, et cetera. And so that was really the key differentiator for us, is that you could, again, apply the right sequencing technology to the problem at hand. Because as you may or may not be aware, not all of these platforms perform exactly the same. There are some benefits to using platform A as opposed to platform B, and there isn't one clear winner in every circumstance. It depends on what your sample type is, the sorts of genes that you want to analyze, et cetera. And so to us, that was incredibly important to us. And so we could provide that customizable service, deep experience with formalin-fixed paraffin-embedded tissue to pick the right methodologies for processing the sample, as well as the right platforms for executing the sample, and then the correct bioinformatics pipeline to interpret the results that come out. And so, as you all know, this is incredibly important because there's a growing number of therapeutic targets. We have smaller and smaller sample sizes. We want to get more detailed information out of the samples that we have. And to do that, you have to be able to get data, and that data has to be accurate and reproducible. And so that's really what we're leveraging through our genomic laboratories at Seqrite. In addition to that, there has to be some quality program around it, because you want to take the results that you've got, and if you're going to use them in clinical trials or move those forward to the FDA, you have to have done that in a robust and a compliant mechanism. As you know, a lot of the next-gen things that are happening now are happening in postdoc laboratories, and they don't have that same rigor of compliance and CLIA certification around it that's really necessary to move forward, at least to the regulatory agencies. So as you know, there's many advantages to next-gen. The best advantages are really that you get the breadth of Sanger sequencing, but with the added sensitivity, because you can add depth to your profiling. You can look at many genes at the same time. You can look at only hotspots or provide as broad coverage as you want. So you can basically formulate the question that you want to ask up front, and then it's possible to design that assay with the technology and address that particular problem. But there's still issues. So I'm going to use an example in colon cancer and lung cancer, because this has been a little bit better studied. So there's an antibody drug to EGFR that's commonly used in colon cancers, two versions of it, cetuximab and panitumumab, one from BMS, one from Amgen, that actually predict non-responsiveness to that anti-EGFR therapy if you have a mutation in the KRAS gene. And we posited all the reasons behind that. The KRAS is downstream from EGFR, and so if you have a constitutively activating mutation in the KRAS gene, then putting a block above it probably wouldn't provide much benefit, and that seemed like a reasonable hypothesis at the time. You know, there was questions of when we detect a mutation, is it in one cell, is it in all the cells? Do passenger mutations that are also present in those cells affect that KRAS mutation? What's the influence of surrounding stroma? We've seen lots of talks here on tumor-infiltrating lymphocytes and how they might affect things. So there's lots of unanswered questions around it. But more importantly, when cetuximab and panitumumab were placed in lung cancer trials, it turned out that the presence of the KRAS mutation didn't predict non-responsiveness to the drug. The drug worked just fine. So that raises the questions of just identifying a mutation just because it was actionable in one organ does not mean that it's actionable in another organ. So that further complicates the issue of, should we just be broadly looking for a wide variety of genes and then understand how to act on those? We may or may not. So the service portfolio that we're currently offering at Seqrite really focuses on research services that are regulated, highly regulated, CLIA environments, with the full rigor of GE behind it. And then biopharm services, we're really looking to expand out and figure out what these mutations mean in the current environments. So, sort of giving you the overview, I'm now going to change directions a little bit to a new technology that was initially developed at GE Global Research and more recently actually implemented in our Clarient laboratory. And the real question with this was, how do we take a single piece of tissue and extract all the information that we want to get out of it, understanding that our current methodologies of just doing genomic analysis and expression analysis tell us about tissue that's been ground up. So what if the interrelationships of cells make a difference? Does it really make a difference whether cell A is doing one thing and cell B is doing something else? And I'm going to show you some data around that. So we basically invented a methodology that replaces what we currently do, which is we get a single section of a slide today off of our paraffin embedded block, and we perform one stain on that section. So we've got our cell of interest. Typically, we find something on an H&E. We hope that our next cut into the block has the same overall representation of tumor that we saw in that H&E cut before it. And then we stain for a one-protein biomarker, and then we interpret that in the context of the H&E. What multiomics does is it allows us to repeat almost as many stains as we want on that single slide. So you're not limited to one stain on one slide, and then you cut deeper in the block, which is actually different cells, and then assess the protein level in those cells. This is on exactly the same cells. You can quantify as many as 60+ proteins all simultaneously. And the way that that is done is through a process of staining, scanning the slide, inactivating the stain, restaining, and rescanning the slide. So it's an interactive process that involves digital image analysis on exactly the same section in a repetitive sequence. So what you wind up getting is a multiplex stain in which you can look at multiple markers on exactly the same section of tissue. And of course, the viewing software and how you interrogate that is going to be the next big step, and I'm going to show you some interesting slides around that. So what does it mean that you can do? It means that you can look at all of these different proteins simultaneously on the same slide, but when you get to the end, you can also apply in situ hybridization markers. So that means that not only can you do this multiplex within proteins in immunohistochemistry, you can also extend FISH probes in exactly the same cells. So you can start talking about DNA copy number, translocations, amplifications, deletions, along with protein analysis in the same sections. In addition to that, there was still the problem of if you took an H&E and then you apply these markers, you're applying the markers to the cut that wasn't exactly your H&E. They're slightly different cells. They're in the same area, but they're still a couple of microns deeper. And so we were managed to be able to take these stains and create a virtual H&E. So the slides that you're looking at on screen aren't actually an H&E. They're a computer-derived H&E from the individual stains that we performed as part of the 60-plex in this example. And we've used this example further to take a look at several adenocarcinomas that look roughly similar but behave quite differently. And so if you take, for example, these are molecules AKT, c-Met, ERK, and phospho-ERK, and we apply it to those three cancers, you can see the expression of those pathway markers are entirely different between the three cancers. So that's sort of where I was going to when I showed the mammoth under the microscope. We can do pretty well looking at morphology under the microscope, but many things that look similar to us under the microscope have wildly different biology when we look at it. Now, the problem is, can you grind up that tissue and see these same sorts of events? I can tell you, it becomes difficult. So one of the things that we can do with this is within the same exact section, I'm going to show you the sort of the plethora of stains that can be done. So you can see the many stains that were done here, but within those same sections, even more stains were done on top of those, and even more stains were done on top of those. So in our sequence of how this works, each one of those images were stained, scanned, destained, scanned again with the next two markers, destained, et cetera, in that repetitive process. So this process that you saw here took a repetition of 12 or 14 stain and erase processes to get the 2 dozen or so stains that we could analyze then with bioinformatics. So there's going to be a poster on this presented tomorrow morning in which 25 biomarkers were done on 800 breast cancer cases. Really shows the power of what you might be able to do with it. It wasn't done to really do the full analysis of, and here's how we treat patients. This was done more as a feasibility of, here's what you can actually apply, and if you have patient cohorts with outcome, you can start addressing very, very complex questions with technology that exists today. So if we look at what we've done, we've actually assessed over 200 antibodies, and if we took Hanahan and Weinberg's Hallmark of Cancer article and we overlaid where those 200 antibodies fall, you can see that we cover all of the hallmarks of cancer. So it's not comprehensive to every known protein, but if you had questions about specific pathways, you could address them very nicely with what has been assessed thus far. The sample analysis workflow is sort of as presented here. We do have to conjugate these antibodies. It is a proprietary process for doing the conjugation. Almost all antibodies conjugate, but not all of them. There are some antibodies that just don't conjugate with the process. For the most part, the order in which you apply the stains does not make a difference. So it doesn't make a difference whether you drop the antibody first or you drop the antibody 50th. You get the same results. That's true for about 95% of the antibodies that have been validated to this point, but with about 5% of the antibodies, the order does make a difference, so we have to do the experiment to make sure that the order does or does not play a difference. If it does, then the appropriate antibodies need to be moved early on in the process. And if we're doing whole slide analysis, we have to decide which regions of the tumor we're going to do this deeper analysis on, because you can imagine the amount of bioinformatics data that you get back from this can be tremendous. Publications that you'll see in the back of the room, most of the slides that I've showed you today have come from the PNAS publication in July of this year. It details all of the methodology, goes through all of the processes that were involved within preparing this, as well as several other recent publications on the technology. One of the tricks to this as a pathologist is how do you visualize the data? So you get this wave of information. It's a little bit like next-gen sequencing. You get this wave of information that comes in. You go, well, how do I make sense out of it? I'm like seeing all sorts of things. And so one of the things that we recognize early on is we need a process so the pathologist can actually view what we're doing with all of these stains. And you can do lots of different things with them. So you can just overlay different colors, so you can make fluorescent stains look like chromogenic stains. We could create heat maps, so you can combine multiple antibodies together to talk about as a PI3K pathway activated, for example, and do a heat map to say it's on, it's off, or it's neutral. You can build interactive modeling with it, so you can sit down with the slide and ask what-if questions and make the stains almost magically appear in front of you to say, show me this but not that. So all of these tools are being developed right now, and it can be fairly complex. In addition to this, by applying what we call segmentation markers, stains that we know only stain within the nucleus or only on the membrane or only within the cytoplasm or only in specific cell types, we can use those markers to fully segment whether the staining is specifically on a membrane. So, you know, if you look at a HER2 stain and you want to quantify the amount of membrane staining, it becomes difficult because you also get some cytoplasmic staining in some cases. So being able to see what's there is difficult for the human eye to do. Using these subset markers, we can localize the stain to even specific organelles within the cell. So you're just sort of overlaying, and then that will tell you where the end results actually are. In the end, what you're really getting is a massive spreadsheet on a cell-by-cell basis. That's actually what you're getting. So you say, I got a cell. Here's the coordinate of the cell. I understand the spatial relationship of this cell to all other cells in my mix. For each of those cells, I understand the staining, location, and intensity of any of the markers that I've applied. And given that you've got all this data, you can do the same sort of clustering analysis that we wind up doing with either gene expression data or with next-gen data. And you could take that clustering and you can, tell subclasses and give a variety of different percentages of things. But the key is that you can apply it back to the original cells. So that's where the pathologist interacting with the slide becomes very important, because you can ask questions and you can start asking heterogeneity questions. So here's a great example. This is from a colon cancer case. So got a variety of different drugs that work on the mTOR pathway. And if you did, for example, an expression analysis, you would get a result that would just say, well, I believe that the mTOR pathway is activated, or I believe that it's not activated. And of course, you wouldn't just be looking at tumor cells. You'd have all the stromal cells and inflammatory cells and everything else that you mixed in between. This question, we looked at a couple of downstream markers, S6 and one of the 4E proteins downstream of mTOR, and asked the question, say, well, sometimes when that mTOR pathway is activated, both pathways are turned on. Sometimes when it's activated, the S6 pathway is turned off, but the 4E pathway is turned on, and sometimes it's vice versa. And we can color code that, and that's exactly what we did in each of these examples. And you can see in many of these examples, cell-by-cell expression is different. So if you'd used a bind and grind assay to just put it all together, you could actually come up with the wrong result. So you can actually have an S6 pathway active, a 4E pathway active in just some percentage of the cells, but if you ground them all up and just looked at the blend, you might not be able to see that. You certainly wouldn't be able to differentiate the cases in which there's no heterogeneity from those cases with significant heterogeneity. So we believe this is going to become incredibly important to understand the nuances of what our drugs are doing and how to better target individual patients. The cluster analysis really does demonstrate that large-scale divergence on a cell-by-cell basis. And I believe that's the news of this technology, is that what we've thought about in blending studies actually has a lot more complexity buried underneath it. Now, for the first time, we can see that, and it's really just a question of building the tools and the resources around it to understand how we best interact with it. And that's the tool sets that we're building, and we have a variety of technologies built around that. So we can either co-localize different markers or overlay markers on top of each other with the software currently, and we've tried to apply this clinically. Our clinical example was actually Hodgkin's disease, because Reed-Sternberg cells are very rare. We want to look at a co-expression of a wide variety of number of markers on very, very rare cells. So for us as a platform clinical challenge, represent a good route to take. And in essence, what we had to develop first was the ability to take bright field IHC, which pathologists were used to interpreting, and change those fluorescent signals to look like bright field images. So the images on the top and the bottom, one are real images and the other are pseudo-derived images from the fluorescent stains. And then the second thing was to be able to develop markers so that, or viewing platforms, so that the pathologists could actually interpret the slides. Now, the good news is telepathology is here, and most pathologists are very comfortable with telepathology systems, so they were already comfortable in driving a slide around on a monitor. It was merely creating the tool sets that allowed them to look at multiple stains at the same time. This is the one that our particular hematopathologist really liked and developed, and it was an ability to take a single cell and do what we call window tiling. So on the left-hand side of the slide is a CD30 stain, on the right-hand side of the slide is a CD45 stain, and they can merely take it like a window pane and drag it back and forth to see whether that cell is expressing one marker or another. And so this is now just tiling vertically instead, the CD30 on top, the CD45 on the bottom, and you could just move that window up and look at the opposite of that same staining pattern. So again, one of many visualization tools, but if you wanted to look at all of the stains simultaneously, you could set up systems to do that too and say, here's the cell of interest, show me how all of the markers are expressing on that cell. So really the offerings around this, I think, are the most exciting offerings. So the issue around this is, even if you have just a single slide available from a study, you can come up and choose from the variety of 200 plus markers that have been assessed. We create the appropriate ordering for those antibodies, and then those can be executed on those individual slides. And it just takes the one H&E section. So even if you have very, very limited study samples, it's not like, oh, I can only answer this one question. If you have a study out there and you actually want to assess 4 or 5 different pathways potentially, you don't have to make a choice of, I'm going to pick one protein in one pathway. You can assess them all simultaneously. In addition to that, we're also very open to doing biomarker development. So if you have your own antibodies or your own pathways that you would like to be delved deeper, that's just a more detailed analysis for us to say, can we get those antibodies to conjugate appropriately and then integrate them into the commercial offering. Got a website for this called www.multiomics.com. You can just go to that website and it'll give you lots of additional information on that. So our real challenge in all of this when we started Clarient was really trying to find the differences that make a difference. I think these technologies are where it's at. We've provided the expertise in breast pathology to help answer those difficult day-to-day questions in pathology today, but we've also extended out the services so that you can have the best of the next-gen technologies, whether it's next-gen sequencing or it's whether these true next-gen tools of really being able to localize which cells are doing what within a tumor. And this fits within, this is my last slide, but this fits overall within the GE vision of precision medicine, of really trying to understand each patient from a physiology level to a risk factor level to the types of disease level. Have tools across our continuum of care from screening in our in vivo sections through diagnosis, staging, treatment planning and monitoring, and that includes all of the in vitro work that I talked about today, as well as some of the newer molecular probes like the PET probes that are coming down the line in the future, as well as some of our life sciences products possibly for disease monitoring in the future. And it means that we've got lots of areas of focus as a company so that we can have a broader breadth. Again, it's having the plethora of resources at hand so you can use the right tools to address the problem appropriately. So I am going to end there. I'll open it up to any questions that anybody has. And like I said, I don't know how we're doing on time, but if we're running too late, I'll hang around for a while, free to answer anybody's questions. Thank you, and we'll open it up for any issues. Not if you're embarrassed, I'll be up here, so feel free to come up and ask individually. You have a question? Yep.
So what type of material can you use? Can you use this technique on archival H&E sections and archival immunohistochemistry slides?
So we have not done that. We can use it on archival formalin fixed paraffin embedded material, certainly. But the first process requires a destaining. I don't think, I refer it to some of my research colleagues, but I don't think that you can start out with a previously stained slide, especially in H&E that has, because it's a fluorescent architecture, so the eosin is, you know, going to background fluoresce like crazy.
And have you tried it on core biopsies?
So yes. So it's been done on core biopsies. It's been done on TMAs for research purposes. So large-scale TMAs, it's actually a lot of the work has been done on TMAs. But yes, core biopsies, hold sections, even FNA pellets. You know, one of the key things for this, it's been done on CTCs, correct? So yes, one of the key things with this clinically, it's not a problem in breast cancer today, but when you have limited tissue, like lung cancers, this could be very critical because we just want to run standard markers and you don't have enough sections to actually execute that. This might be a big deal in breast as we move to reassessing metastatic lesions, right? We've got a dictum now, and we understand that primaries of metastasis don't always show the same levels of expression, whether it's at a protein level or at a genomic level. Well, while we have lots of access to the tissue primary sample, because they're all excised, in a metastatic setting, you might only have an FNA sample. And then that limited sample, this might be the optimal technique to address all of the questions of the day. Yes? I'm sorry, can you go to Mike, just because I can't?
Clinically approved.
So, correct. So the only thing that we've launched clinically through our CLIA laboratory has been the Hodgkin's profile. We've performed dozens of these clinically, mostly because there were small samples. Patient would have had to undergo rebiopsy without this technology. And we're working with the insurance companies now. So yes, we have had some insurers that have looked at the technology, said this has saved us significant dollars and have paid. But we have not, the technology is too new where we don't have a systematic approval from the insurers that say anytime you use this technology, you will get paid. We haven't been rejected from anybody, I should put it that way. So it's either in the process or somebody is approved.
What about reimbursement through Medicare?
So again, we haven't submitted formally through that process yet. The question with all of these things is going to be what's the indication for use? Clinically, our current indication for use is when the sample size is so small and the clinical question so important that the patient would undergo rebiopsy if we can't get an answer with this technology. So there's no way to answer the question with the current sample. And because you can't answer the current question, the patient's going to undergo some procedure to acquire more tissue to address that question. And we're saying in that circumstance, hold the phone. We might be able to answer the question with what we already have.
Thanks.
I have a question.
Yes.
How long does the process take? You said that, you know, you stain, destain the same section. I mean, what about the fact that the sections can detach from the slide?
So, you know, that's one of the issues right now, keeps on shortening. The actual longest period of time is actually the scanning, not the staining.
Not the staining.
So the staining and destaining process are actually relatively rapid,